Hey guys, this is me Akash here for Biology Vibe. So today we're going to start Vibe 1.7 named Transcription. So basically transcription is what? Transcription is basically when the DNA that is sitting inside the nucleus sends it codes via processing an mRNA, that is the messenger RNA. So transcription is basically to know the process how DNA makes that mRNA, that is the messenger RNA. As the definition says, transcription is the process by which the information in a strand of DNA is copied into a new molecule of messenger RNA. So this is your DNA molecule, this is your RNA polymerase, and a new molecule of mRNA is being synthesized. The sequence in which the bases are arranged in this mRNA is complementary to what it is arranged in the DNA. So this is a kind of block diagram which shows what happens in transcription, how it happens. So this is 3' end, this is 5' end. So the RNA polymerase has the ability to synthesize the mRNA in the 5' to 3' direction. So the components of this is, one strand is called the template strand, this blue one, or the antisense strand. One strand is green one, that is the coding strand or the sense strand. There's a promoter and there's a terminator. Promoter from where the transcription that's the transcription start site, from where the transcription will begin, and at the terminator it will end. So the basic is the RNA polymerase binds to the template strand and synthesizes from the promoter till the terminator and keeps on adding the sequence which is complementary to the template strand. The template strand is called the antisense strand and the coding strand is called the sense strand. Reason being, the template strand, the complementary sequence of the template strand is there in the coding strand, and a new mRNA will be formed keeping template the main thing, and the bases which will be added in the new mRNA will be complementary to this template strand. So if you see, the base sequences in the mRNA and the base sequences in the coding strand will be the same. That's why the coding strand is called the sense strand and the template strand is called the antisense strand. It moves from 5' to 3' direction, keeping this blue part, that is the template strand, as the base, and then nucleotides are added, that is complementary to this template strand. So it has three main phases: initiation, elongation, and termination. Let's see what happens in the initiation phase. Basically, what happens is like there's a factor known as the sigma factor that recognizes that promoter site from where the transcription has to be started. It recognizes the promoter site of the DNA and helps the attachment of RNA polymerase. Then definitely it has to transcribe it. Then the hydrogen bonds have to be broken. It is broken by the RNA polymerase. The template strand takes part in transcription, the main one, that is the baseline, and the complementary strands need to be added. The transcription proceeds in 5' to 3' direction. Pyrophosphatase hydrolyzes two phosphates from the activated nucleotide to release energy needed in the process. So basically it's an energy dependent process. So these are the enzymes which hydrolyzes two phosphates and gives the process the essential energy. So this is the RNA polymerase and the sigma factor. The sigma factor helps to recognize the promoter site. That means the site from where the transcription will start. That is initiate. Now the part is elongation. This yellow part, that is the messenger's mRNA, will be extended and elongated from this part. RNA polymerase establishes phosphodiester bond between adjacent nucleotides. That means the synthesis of RNA will be started. That is elongation. In the elongation phase, it will be extended. In the elongation phase, this sigma factor will separate from the RNA polymerase coenzyme. Sigma factor separates from the core enzymes, moves along a template, that is the antisense strand, till it reaches the terminator site. So initiation is done, elongation done, and the third part is termination. Now let's see how the termination occurs. When this RNA polymerase get to know that yes, it's the end and the mRNA needs to be released from here. The RNA polymerase enzyme reaches the terminator sites. There's a specific site known as the terminator site. At the terminator site, it separates from the DNA template. On terminator sites of DNA, nitrogenous base pairs are palindromic. Palindromic means it's the same from the either side. For example, if it's A, G, T, G, A, if you read from forward or from the backward, it's the same. This is known as palindromic site. So when this RNA polymerase reaches a site that's palindromic, it senses that yes, it's the time to terminate. RNA polymerase recognizes the terminator sites and stop the synthesis. Okay, so in prokaryotes, there's a specific protein which is known as Rho protein that is responsible for this termination. So in case of prokaryotes, Rho factor comes into picture, and then RNA polymerase signals that yes, it's time to terminate and release the mRNA. mRNA comes out from the nucleus and moves towards the protein factories of the cell, that is ribosome, where the translation will occur. So this is a kind of a whole picture which what happens when in initiation. There's this yellow portion is a sigma factor, there's a start site, and this blue part is known as the RNA polymerase. It will start. This yellow part is the gene which is to be synthesized. This lower part is template. The bonds will start, means the hydrogen bonds will break, and in the elongation phase the sigma factor will be released and the mRNA will be synthesized, keeping this lower part as a template, adding complementary sequences. And then it reaches the terminator site. Okay, at the terminator site it's palindromic. In case of prokaryotes, certain proteins are there, Rho proteins, that will signal that it's the part to be terminated, and this blue chain, that is the mRNA, will be released and it will go to the nucleus for further translation. So transcription process is over, and now this mRNA has to go to ribosomes for translation. So in the next vibe, that is vibe 1.8, we'll discuss about translation. So if you like the video, please hit the like button, share this video with your friends, and definitely subscribe to my channel to get more and more videos like that.